Good morning, everyone. Today, we are going to delve into reviewing past A-level examination paper. This session is designed to help you understand how to approach this paper effectively and ultimately enhance your exam performance. Without further ado, let's get started. The diagram shows the ultrastructure of a typical animal cells, which structure synthesis and transport lipid. The correct answer is C, which is smooth endoplasmic reticulum, which is responsible for the synthesis and transport of lipids. A light microscope is used to observe two structures that are 200 nanometer apart. And how far apart are the structures when the magnification is changed from multiply 40 to multiply 400? To find the distance between the structures at different magnification, use this formula. Distance at new magnifications is equivalent to original distance divided by new magnification factors. Given in the questions, original distance is 200 nanometer, original magnifications is multiply 400, and new magnifications multiply 400. And the magnification factors that is new magnification factors equals to 400 divided by 40, you get the value of 10. And now calculate the distance at multiply 400. Distance at multiply 400 is equivalent to 200 nanometer divided by 10 is equal to 20 nanometer. And 20 nanometer is equivalent to 20 micrometer. So the answer is B. The diagram shows a stage micrometer scale view through an eyepiece containing a graticle. The small divisions of a stage micrometer scales are 0.1 mm. The stage micrometer scale is replaced by a slide of a plant cell. This question is a similar question appeared in October 2020. What is the length of the nucleus in the plant cells? The answer is 25 micrometer. Some features of cells are listed: cytoplasm, cell surface membrane, ribosomes. Which features are found in both animals and prokaryotic cells? The features found in both animal cells and prokaryotic cells are cytoplasms, which exists present in both animal cells and prokaryotic cells. Cell surface membrane is also present in both, and ribosomes is present in both, although they differ in size and structure. The answer is 1, 2, 3. Which size of ribosomes is found in chloroplast? This is also a similar question appeared in March 2021. The correct answer for these questions is B, 70S. Typical plant cells and prokaryotic cells both have 70S ribosomes. Plant cells also have 80 ribosomes, but they have 70S ribosome in their chloroplast and mitochondria. Prokaryotic cells primarily have 70S ribosomes. And centrolus, lysosome, and cellular cells do not fit the given presence or absence criteria for both cells type as per the other options. In which of these organelle in ATP synthesis? ATP is synthesized in chloroplast and mitochondria. A sample of milk is tested with betadine solution. After boiling, a yellow color is observed. Which conclusion is correct? The correct conclusion is D, reducing sugars are present. The yellow color indicates that the reducing sugar, such as glucose, is present in the sample. The table shows some information about carbohydrate polymers, which row describe amylose. The answer for it is B. And which row about alpha glucose and beta glucose molecule is correct? The answer is A. Some of the molecules found in animal tissues are grouped into 3 lists. The correct answer is glycogen antibodies, adenine, phospholipids, and hemoglobin, carbon dioxide, mRNA, and monosaccharides. Both list 2 glycogen antibodies and the 9 phospholipid, and 3 hemoglobin, carbon dioxide, mRNA, monosaccharides contain molecules that include nitrogen atom, example antibodies, adenine, hemoglobin, mRNA. Hemoglobin consists of 2 change, 2 alpha change, and 2 beta change. Approximately 5% of all humans have one amino acid in the beta change that has been changed, affecting the structure and stability of hemoglobin. Which of this level of protein structure could be changed in the hemoglobin of this human? The answer is primary, secondary, tertiary, and quaternary. A change in the amino acid sequence primary structure can affect the higher level of protein structure, including secondary, tertiary, and quaternary structure. Which will give the correct description both a collagen molecule and a collagen fiber. The correct description of both a collagen molecule and collagen fiber is collagen molecule is a 3 helical polypeptide change forming a triple helix held together by hydrogen bonds, and collagen fiber is a molecule of collagen lying parallel and cross-linked to each other. The diagram shows the structures of 2 amino acids, each of which has 2 amine group. A peptide bond is formed between the 2 amino acids, which group could form the peptide bond. The answer is 1 and 5. An enzyme was added to a small excess of its substrate. All variables were kept constant. The student was asked to catch a graph to show how the concentration of the enzyme substrate complex changes over time. Which graph shows this correctly? The answer is D. The table contains results recorded by a student from an investigation into the effect of temperature on an enzyme catalyzed reactions. All other variables were standardized. The correct conclusion is 40 Celsius was the optimum temperature, and this is supported by the highest rate of reaction recorded at that temperature. What is the correct range of measurements for the width of the cell surface membrane? And the correct range of measurement for the width of the cell surface membrane is between 5 to 10 nanometer. The cells in the root of beetroot plant contain a red pigment, and when pieces of root tissue are soaked in a cold water, some of the red pigment leak out of the cells into the water. An experiment was carried out to investigate the effect of temperature on the loss of red pigment from the root cells. It was found that the higher the temperature of the water, the higher the rate of loss of red pigment from the root cells. Which of these statements could explain this trend? Statement 2 and 3. Statement 2 is, as the temperature increases, the tertiary structure of protein molecule in the cell surface membrane changes, increasing the permeability of the membrane. And statement 3, phospholipid molecule gain kinetic energy as temperature rises, increasing the fluidity of the phospholipid bilayer and allowing pigment molecule to diffuse out more easily. The cell surface membrane structure is described as a fluid mosaic. The statements that describe the mosaic part of the cell surface membrane is the scattering of the different protein within the phospholipid bilayer. The diagram shows 2 pathways X and Y through which molecule can diffuse across a cell surface membrane, which will correctly show a possible pathway for lipid water and glucose. The answer is A. Some stem cells divide and give rise to phagocytes. Where is the human body do these stem cells divide? The correct answer is C, which is 2 only. Stem cells that give rise to phagocyte primarily divide in the bone marrow. What is the correct sequence of stages in the mitotic cell cycle? The correct sequence of stages in the mitotic cell cycles is C, G1 to S, G2, to mitosis and cytokinesis. The enzyme telomerase repair telomeres. It stops the telomeres from getting shorter each time the chromosome is replicated. Telomerase is not normally active in human body cells, but in some diseases, telomerase can be activated. In which disease is the enzyme telomerase activated? The enzyme telomerase is activated in cancer. In many cancer cells, telomerase activity increases, which allow the cells to replicate indefinitely. The statement described even during the mitotic cell cycle. The correct order of this event in the mitotic cell cycle is 3, 2, and 4, which is 3, chromosome condense and nuclear membranes disappear, and chromosome arrange themselves at the equator of the spindle, and finally, they have a centromeres divide. And this sequence is already explained just now. Which statement about nitrogenous bases is correct? The correct statement about nitrogenous bases is cytosine is a purine with a double ring. Uracil is a pyrimidine with a single ring structure. The other statements are incorrect regarding the classifications of the bases. Rifampicin is an antibiotic used to treat tuberculosis, and it works by inhibiting RNA polymerase in bacteria. Which of these processes will be directly inhibited by the antibiotic? So this is also a similar question which is appear in March 2019. The answer is rifampicin inhibit translation. Rifampicin directly inhibit transcription by binding to bacterial RNA polymerase, preventing it from synthesizing RNA from DNA template, and it does not directly affect DNA replications or ATP synthesis. Melsons and Stas investigated DNA in bacteria. They grew bacteria in a medium with only heavy nitrogen and 15 until all of the bacteria DNA contained only heavy nitrogen. These bacteria were then moved from the heavy nitrogen medium and cultured in a medium with only light nitrogen and 14. Some bacteria were collected from each of the next 3 generations, and their DNA was analyzed. Hybrid DNA contained both heavy and light nitrogen, which row show the correct DNA of the first and the 3rd generation. The answer is B. DNA of first generation is 1 hybrid, and DNA of 3rd generation is 1/4 hybrid and 3/4 light. The diagram shows the stages in the productions of part of a polypeptide. From DNA nucleotide sequence template strand to mRNA sequence and amino acid sequence. Which features of the triplet code is illustrated by the formations given? The features of the triplet code illustrated by the information given is the triplet code is non-overlapping and is only read in one direction. And this is because the mRNA is transcribed in a sequential manner and each triplet codon is read separately without overlapping. Which combination of features is characteristic of a folium sieve tube element as it unloads into a seam? The correct combination of features characteristic of a folium sieve tube element as it unloads into a seam is water potential is indicate low density, and lignification of the cell wall indicate absent. And in this context, the sieve tube have a lower water potential compared to the seam, which allow for the uploading of solutes, and they are not lignified, which is typically for folium tissue. Which statement correctly describe transport pathway in dicotyledonous plant? The correct statement that describe transport pathway in dicotyledonous plants are: in the simplest pathway, water moves through the cytoplasm of cells and not through intercellular space. And 4 is the apoplast pathway may be blocked by a capestrian strip, which forces water to move into the simplest at the endodermis. The diagram shows transverse sections of part of a plant which labor structure transport mineral iron. The correct answer for which labor structure transport mineral iron is C. In this case, xylems 1 and 4 transport mineral iron, while folium 2 and 3 primarily transport organic nutrient. Question 31 is a similar question appeared in October 2017. Which features of transport in plants is correct for both xylem and folium? The vessels that carry fluid by mass flows are, it occurs by mass flow, and these are the folium sieve tube and the xylem vessel. In plants, mass flow occurs in the folium for the transport of nutrient and in xylem for the movement of water and minerals. Arteries and veins in animals primarily use pressure-driven flow rather than mass flow. What is the main function of a companion cells in a folium tissue? The answer for number 32 is providing cytoplasmic contact with the sieve tube element for loading. And the companion cell assists in the transport of nutrient and facilitate the movement of sugar and other organic compounds in the folium. The contraction of the heart is coordinated through electrical impulses passing through the cardiac muscles. What is the correct order of part of the sequence of the impulses? This is also a similar question appeared in May 2017. The question is, what is the correct order of part of the sequence of these impulses? The correct order of the sequence of electrical impulses that coordinated the contractions of the heart is right and left atria to atrioventricular node, and then Purkinje tissue and ventricular walls. And this sequence ensures that the electrical impulses first stimulate the atria to contract, then travel to the atrioventricular nodes, and finally propagate through the Purkinje fibers in the ventricular walls to coordinate ventricular contraction. As blood flow from an artery to a vein, the thickness of the wall of the vessel changes. Which bar chart shows these changes correctly? The answer is D. The graph shows the oxygen dissociation curve of hemoglobin from two species of mammal, S and T. Which statement could explain the difference in the oxygen dissociation curve of species S and species T? The answer is statement 2. The hemoglobin in species T has a lower affinity for oxygen than the hemoglobin in species S. Which tissue may be found in bronchioles? The correct answer is ciliated epithelium, elastic fiber, and smooth muscle. Bronchioles primarily contain ciliated epithelium for mucus clearance, elastic fiber for flexibility, and smooth muscle for regulating airflow. Which of these statements could describe the effect of carbon monoxide in cigarette smoke? The answer is it binds irreversibly to hemoglobin, which reduces its ability to carry oxygen, while it can affect the respiratory system and cardiovascular function. Option statement 2, 3, and statement 4 do not accurately describe the direct effect of carbon monoxide specifically. The global mortality features of some diseases in 2002 are shown in the table. How many millions of people died from viral diseases listed in the table in 2002? The answer is B, 3.4 millions. And which of the statements could describe both B lymphocyte and T lymphocyte? The correct answer is they contain specific protein receptor in their cell surface membrane. And number 3, statement 3 is they divide by mitosis. Let's look into the explanation. Both B lymphocyte and T lymphocyte contain specific protein receptors in their cell's membrane and can divide by mitosis, but only B lymphocyte differentiate into plasma cells. 2 people G and H were even given an injection to protect them against a particular pathogen. One person was injected with antibiotics, and the other person was injected with a vaccine. So the graph showed the concentrations of the antibody against this pathogen in the blood of the 2 people G and H over a period of 20 days following the injection, and which will correctly describe the types of immunity shown by G and H. G is artificial active immunity and H is artificial passive immunity. That's all for today's presentation. Don't forget to hit the like button, turn on your notifications to stay updated, and please subscribe to Jom Study Lab YouTube channel. Thank you very much for your operations.